Hello, everyone, and welcome to our discussion about microbiology. First, let's review the characteristics of your bacteria. Bacteria are prokaryotic, they contain both DNA and RNA, and they multiply by binary fission. Bacteria are often measured in micrometers, and their average size ranges from zero point 0.2 to zero point 0.4 micrometers, and they produce either exotoxins and endotoxins. So, first, let's differentiate an exotoxin from an endotoxin. Exotoxins are produced by both gram-negative and gram-positive bacteria. They are protein in nature and they are excreted by a living bacterial cell. All gram-positive bacteria produces exotoxin except for Listeria. Take note of this one. And some exotoxins are in the form of enzymes as well. For example, we have the coagulase, and below are some of the common exotoxins. Number one, we have the cytotoxin that kills host cells, enterotoxin that damages the cells of the GIT, and the neurotoxin that interferes with the nerve cells impulses. Meanwhile, an endotoxin are produced only by gram-negative bacteria. It is a part of the outer membrane of lipopolysaccharides, and they require cell death for release, and their effects are more devastating. So unlike exotoxin, it is not protein in nature. And guys, I want you to remember this test as well. We have the Lamulus lysate test. This is a test to detect endotoxins in body fluid, and the positive reaction or the positive result here is the clumping or agglutination. And this test makes use of an aqueous extract of blood cells from a horseshoe crab. Let's also review the parts of your bacteria. We have the first one, the glycocalyx, and found there is our capsule. Capsule are organized and firmly attached. They are also antiphagocytic. It is composed of polysaccharide and a polypeptide protein. Remember this test? The Neufeld-Kellogg's test. This is a test for capsule. And some of the bacteria that have capsule here, we have the Streptococcus pneumoniae, Neisseria meningitidis, Klebsiella pneumoniae, Haemophilus influenzae, Salmonella typhi, and Bacillus anthracis. I want you to remember the Bacillus anthracis. It has the poly-D-glutamic acid capsule, which is not a true capsule, and it is stained by the McFadden's stain. The next one, we have the slime layer, which is unorganized and not firmly attached, and it is seen in Staphylococcus epidermidis, which is a biofilm. The next one, we have the cell wall, and it gives shape to the bacteria. Peptidoglycan is its main component, and it's the basis of gram stain, specifically the murine layer. So always remember that gram positive with rigid peptidoglycan layer, while gram negative have thinner peptidoglycan layer, and it is also the site of antibiotic action. For example, we have the penicillin. I want you to remember as well that M protein found in Streptococcus pyogenes prevents phagocytosis, and mycolic acid found in Mycobacterium species prevents digestion of the bacteria during phagocytosis. And also, Mycoplasma and Ureaplasma have no cell wall and are not considered as true bacteria. Number three, we have the plasma membrane or the cell membrane. It is the site of energy synthesis. The next one, we have the nucleoid, responsible for antibiotic resistance and is transferred through conjugation. So we have two, the chromosomal DNA and the extrachromosomal DNA. So the chromosomal DNA is for sexual reproduction, while the extrachromosomal DNA is a plasmid which carries an antibiotic resistant gene. The next one, we have the ribosomes, which is the site of protein synthesis. And we have next our inclusion bodies, and these are accumulated food or nutrient reserves. So the first one, we have the much granules, which can be found in Mycobacterium tuberculosis. Next one, we have the metachromatic granules, aka the Babes Ernst granules or volutin granules, found in Corynebacterium diphtheriae. And we have the bipolar bodies, or bipolar bodies have safety pin appearance, and they are stained by Wiesen stain, and they are commonly seen in Yersinia pestis, Vibrio parahemolyticus, Burkholderia, Pastorella, Bordetella pertussis, and Francisella. And the last one, we have sulfur granules, which can be found in Nocardia and Actinomyces. Next part is we have the endospores, and they are resistant structure enabling bacteria to withstand adverse conditions. Remember that autoclaving destroys spores. They are made of calcium dipicolinate or dipicolinic acid and pipicolinic acid, and they are commonly stained by Dorner, Ritz-Conklin, and Schaefer-Fulton. And endospores are commonly found in Bacillus and Clostridium species. So, guys, please remember that the spore in Clostridium tetani is found in the terminal part. The Clostridium for Clostridium botulinum, it's in the subterminal part, and for Bacillus anthracis, it's in the central part. The next one, we have the pili or the fimbriae, and they are hair-like projections which is usually found in gram-negative bacteria like E. coli and Neisseria gonorrhoeae. And it has two types. First, we have the somatic, common or ordinary pili, and its function is to adherence to host cell, and this is the first step in establishing infection. And the next one, we have the sex or conjugation pili, and this is mainly for gene transfer and for plasmids. So for the next part, we have the flagella, and it's for locomotion. Many bacilli and spiral are motile, while true motility is seldom observed in cocci, and motility is best seen at room temperature. Always remember that. And eggshell filament, aka periplasmic flagella, are seen in spirochetes. So guys, I want you to remember the classification of bacteria according to flagella because this one are very important. So first, we have the monotrichus. This is a single flagellum at one end, and they are commonly seen in Vibrio and Pseudomonas species. The next one, we have the amphitrichus. It's a single flagellum at both ends, and it's commonly seen in Campylobacter jejuni. And the next one, we have the Lophotrichus, and it's a tough bundle of flagella in one or both ends commonly seen in Helicobacter pylori. Next, we have the peritrichus. This is a kind of flagella that the entire bacteria are surrounded by flagella, and it's commonly seen by most members of Enterobacteriaceae. And the next one, we have the atrichus. It's non-motile or no flagella, commonly seen in Shigella, Klebsiella, Yersinia, Corynebacterium, and yes, Corynebacterium and Corynebacterium diphtheriae. So we have different ways to demonstrate motility. So the first one, we have the hanging drop method, which is the most common. Next, we have the flagellar stains. And the next one, we have the semi-solid media. So guys, please remember that we use tannic acid to precipitate flagella. Okay? Okay, so let's now proceed to the bacteria physiology or requirements for growth. So for the first one, we have the oxygen requirement. So the first one here, we have the obligate aerobes, and they require oxygen for growth. Mainly, they are commonly seen in Pseudomonas, Neisseria, Bordetella, Brucella, Francisella, and Mycobacteria. Meanwhile, obligate anaerobes will die in the presence of oxygen, commonly seen in Bacteroides, Clostridium, Fusobacterium, and Veillonella. Facultative anaerobes, aerobes that can survive with or without oxygen, and this includes most pathogen like E. coli and Staphylococcus aureus. And next, we have the air-tolerant anaerobes. These are anaerobes that doesn't grow well but can survive in the presence of oxygen, and they are commonly found, or they are Lactobacillus and Propionibacterium as the examples. Microaerophiles in the other hand, they are the they required reduced oxygen concentration. And the example here, we have the Campylobacter jejuni and capnophiles in the mean. On the other hand, they require increased CO2 concentration of 5 to 10%, and this includes Neisseria and Hasek group. Next, we have the nutrition, autotrophs and lithotropes, or autotrophs or lithotrophs versus heterotrophs or organotrophs. So autotrophs are inorganic sources of carbon like CO2, and heterotrophs are organic sources of carbon like glucose, proteins, or lipids. So for the next one, we have the temperature, and guys, optimum temperature are best suited for bacterial growth. Okay, so the first one we have here, the psychrophiles. Psychrophiles can tolerate cold enrichment technique for cold-loving bacteria, and both are motile at 25 degrees Celsius but non-motile at body temperature. And we have the examples here. We have the Listeria monocytogenes and Yersinia enterocolytica. Mesophiles, they can grow in temperature of 20 degrees Celsius to 45 degrees Celsius, and they include most pathogens. Next, we have the thermophiles that requires 50, or they grow at 50 to 60 degrees Celsius, and they are called, or they are also called, heat loving bacteria. So we have here the Bacillus stearothermophilus, which is our autoclave's biological indicator, and the Thermus aquaticus, which is the source of DNA polymerase in PCR. The last one, we have the hyperthermophiles, and these are organisms that can grow at 80 to 100 degrees Celsius. For example, we have the Archaebacteria. So for the pH requirement, acidophilic is the term that we use if the bacteria or the organism requires growth at 3.0. And the example here, we have the Lactobacillus as this acidophilus. The neutrophilic, they can grow at pH 6.5 to 7.5 or 7.2 to 7.6. And this includes most pathogens. The last one, we have the basophilic or alkalinophilic, and they can grow at pH 8.0 to 10.0, commonly found in Vibrio species. And also remember, guys, that pH 5.0 to 6.0 is for fungi, or fungi can grow at that kind of pH. The next one, we have the salt concentration, and halophilic organisms require increase concentration for growth. So I have made a table to differentiate each concentration and what bacteria or what organisms can grow there. So for the first one, we have 6.5 NaCl, which can, or where Enterococcus can grow. Next, we have the 7.5% Staphylococcus aureus, and the next one we have 0, 3, 5, 7, 10% for Vibrio cholerae and Vibrio mimicus, and the 5% are for some mycobacteria. We have also here other special requirements. The first one is increased protein for Mycobacterium tuberculosis, and the X and V factor for Haemophilus species. Next, we have the bacterial growth curve. So first, we should learn how bacteria multiplies or how bacteria evolves in our body. So the first phase is we have the lag phase or the adjustment phase. These are bacteria, or in this phase, bacteria are preparing to divide, and there is no cell division, but the bacteria increase in size and not in number. Our next phase is the log phase or the exponential phase, wherein the bacteria numbers increase logarithmically. And the next phase, we have the plateau or stationary phase. In this phase, nutrients are becoming limited and the numbers of bacteria remain constant, although viability may decrease. The last phase, we have the death phase or phase of decline, wherein number of non-viable bacterial cells exceeds the number of viable cells. So remember the causes of bacterial death. Number one, we have the food shortage, the increased concentration of toxin products, and the development of unfavorable pH. Next, we should learn also the correct specimen collection and processing when it comes to microbiology. So the rules are to collect specimens during the acute or inflammatory stage, collect before antibiotic therapy, and the sample should be sufficient. And also the use of aseptic technique and proper medium should be observed, and also the delivery of samples to the laboratory on time. Next, we have the specimen preservation. First, we have the boric acid that may be added to urine to maintain an accurate colony count. Next, we have the zero point 0.5 or 0.025 sodium polyanethyl sulfonate (SBS) that is used to prevent clotting of blood. And always remember, guys, that citrate and EDTA shall never be used in microbiology. And heparin, meanwhile, is used for viral cultures and for isolation of Mycobacterium species. Let's proceed now to the specimen storage. First, we have for urine, stool, sputum, or swabs not for anaerobes, foreign devices such as catheters, and viral specimens can be maintained at refrigerator temperature at 4 degrees Celsius for 24 hours. Remember that CFF, if not processed immediately, can be stored in a 35 degrees Celsius incubator for 6 hours. Serum for serology can be stored for up to 7 days at negative 20 degrees Celsius. I'm sorry, guys, for the typo there. It should be negative 20 degrees Celsius. And tissue specimens for long term storage must be kept at negative 70 degrees Celsius, not negative 700 degrees Celsius. It should be negative 70 degrees Celsius. And specimens most likely to contain anaerobes should not be refrigerated. And we have here, or I got this table from the book of Mahan, and this table is for the specimen storage guideline and which specimen should we refrigerate or which specimen should we leave at room temperature. So I want you guys to read those. And we have here below the holding or transport media, and we have the first one, the Tad Hewitt broth, which is used for Streptococcus agalactiae, the Carrie Blair for stool pathogens, the Ames for respiratory specimen, and we have the Transgrow or Jambec for Neisseria species. Now, let's proceed to our instruments. So the first one, of course, is our incubator. So in our incubator, we use the temperature 35 to 37 degrees Celsius for bacteria and viruses and 25 to 30 degrees Celsius for fungi. Always remember that we need the 18 to 24 hours of incubation for aerobes and 24 to 48 hours for anaerobes. So also here we have our other instruments. We have the Durham tube that is used for gas detection or observation used in water bacteriology. Next, we have our inoculating loop, which is 2 millimeters in diameter that will obtain 0.01 UL of our sample. And our inoculating needle that is not longer than 5 cm, or we should not use inoculating needles that is not longer than 5 cm. And also remember that we don't use dichrome in oxidase tests because this will lead to false positive reaction or false positive result. Next, we have our swab. And for swab, guys, always remember that we use cotton for viruses and we use calcium alginate for Neisseria species. The next one, we have our catheter, and it's acceptable to aspirate urine from the tubing, guys, and not from the bag. Always remember this, that we accept, or it's acceptable to aspirate urine from the tubing and not from the bag in order for us to avoid other organisms or other organisms that may interfere with our culture or with our processing. And next, we have the centrifugation. Always remember that for urine and CSF, we use the 2000 RPM for 10 minutes. And for sputum, we use the 30,000 or 3000 rather RPM for 15 minutes. Next, let's proceed to our clinical specimens. Of course, let's start with blood. So when we collect blood for blood culture, we should cleanse the site with 70 to 95% alcohol and 2% iodine, followed by an alcohol rinse. We have here our 0.025 SPF, and it is the anticoagulant used, but it inhibits some bacteria. So to counteract this one, we should add 1% gelatin. So our dilution factor of blood to medium is 1 is to 10 for children, and 1 is to 5 for adults. And the amount of blood that should be collected is 1 to 0.5 ml or 1 to 5 ml for children and 10 ml for adults. And our routine blood culture in the lab mostly is for 7 days only but for some of the organisms they require long period of time to grow. For example, we have the brucella and the hasek group. They require 21 days or 3 weeks to grow, and for leptospirosis we have 8 weeks in order for it to grow. So our initial incubation medium for blood specimen and is we have the trypticase soy broth rather for aerobes, and we have the thioglycolate medium for both aerobes and anaerobes. Take note, guys, that risazarine dye and methylene blue they are indicators to detect the presence of oxygen. And we have here the signs of growth in the incubation medium. So my mnemonics here is Butch, the Butch. We have bubbles, turbidity, clot or pellicle formation, and hemolysis. So guys, always remember that if positive for growth on initial incubation, always proceed on doing a subculture. Okay? Okay, so the next one we have the CSF, and CSF are usually collected using 3 to 4 tubes. And tube number 2 rather is for microbiology, and it is subjected for gram stain and culture. And if with fourth tube, use the fourth tube for better exclusion of skin contaminants. They are examined immediately or hold either at incubator or at room temperature for no longer than 1 hour, and they are always remembered to never refrigerate in case of delay. And we centrifuge and use the sediments for smears and culture, and we often cultivate it in BAP, MAC, and CAP. And we usually wait for 7 days before discarding the specimen. And we have here the India ink or latex agglutination test that can be performed to detect capsular antigens, namely for Cryptococcus neoformans. And we often collect CSF to detect meningitis. So I have made a table here of the common agents of meningitis and the common age group as well. So first, we have the Streptococcus agalactiae, which is 0 to 1 month, and Haemophilus influenzae 1 month to 5 years old. Neisseria meningitidis that affects 5 years to 29 years old, and Streptococcus pneumoniae, which affects greater than 29 years old. And we have the Listeria monocytogenes for immunocompromised and elderly individuals. Next, let's proceed to our throat and nasopharyngeal swabs. So, most abundant normal flora here is we have the very dense streptococci or alpha hemolytic streptococci. Most common pathogen found in throat and nasopharyngeal swabs, we have the Streptococcus pyogenes or group A Streptococcus or beta hemolytic Streptococcus. And we often culture it on Todd Hewitt broth for fluorescence microscopy of beta streptococci. Always remember, guys, that nasopharyngeal swab is the specimen of choice to detect the carrier state of Neisseria meningitidis and to detect the presence of Bordetella pertussis and Haemophilus influenzae. Next, we have our sputum, and when we collect sputum, we should always note the number of squamous epithelial cells per low power field and PMNs to determine the acceptability of the specimen. And this one is called the Bartlett's qualification or criteria, which requires less than 10 squamous epithelial cells and greater than 25 PMNs. Always remember, guys, that sputum may be often contaminated with normal flora, so that's why we should collect ideally in the morning when it is more concentrated. So for TB culture, specimens must undergo digestion and decontamination. So, our digesting agent acts to liquefy the mucus to free possible trapped bacteria. And we have our gold standard, which is the N-acetyl-L-cysteine or the NALC. And we can also use dithiothrietol. And our decontaminating agent acts to kill non-pathogenic bacteria in order not to interfere with our processing. And our gold standard here is we have our sodium hydroxide. Next, let's proceed to our urine. And midstream clean catch is the specimen of choice when we collect urine samples for culture. Catheterized specimens are for those unable to do a midstream clean catch, and suprapubic aspiration is for anaerobic culture. So, urine must be preserved with boric acid or refrigerated if not processed. So, MAC and BAP are commonly used for culture, and colony count should be performed on all urine samples. So, guys, when we use our calibrated loop, always remember that 1 UL delivers 0.001 ml and the 10 UL delivers 0.01 ml. Also remember that if the colony count is greater than 100,000 CFU per ml, it's significant for UTI or upper or urinary tract infection, and 10,000 to 100,000 is suggestive for UTI, and less than 10,000 is for contaminant, or there's a possibility that there's a contaminant or it's insignificant. Our next one, we have our stool, and it's mainly collected for the detection of enteric pathogens, and if not processed within 2 hours of collection, it should be placed on a transport media like Carrie Blair. And we should always avoid urine upon collection because it is acidic and can kill enterics. Again, we avoid urine upon collection because it is acidic and can kill enterics. So we also use culture, or when we culture, we often use BAP and MAC, or maybe directly plated on a differential media like EMB, and selective media like Hectone enteric agar or HEA, or Salmonella Shigella agar or SSA. And we should always do biochemical testing when we collect or when there's a stool specimen coming in the microbiology department or in the laboratory. So our next one is the genital tract specimen. So for cervix or vagina, when we collect specimen, we should remove mucus before collection. Do not use lubricant on speculum and swab in the cervical canal or vaginal mucosa. Meanwhile, when we collect specimen on the urethra, we should use a flexible swab, and it is inserted 2 to 4 centimeters into urethra for 2 to 3 seconds, or collect or simply collect the discharge. So that's it for our clinical specimens. So for the next part, we should know the ways of studying bacteria. So the first one, it's the study of the morphology and the appearance. So staining is a process of artificially coloring the organism with dyes or stains. So the purpose of staining is to observe and appreciate the appearance of bacteria, to differentiate one organism to the other, and to reveal the chemical nature of the bacteria. So we have our staining or different staining techniques here. The first one, we have the simple staining, which uses simple aqueous solutions of dyes. And the next one, we have the indirect or relief or negative staining, and in this kind of staining, only the background is stained and the organism appears colorless. Our examples here is the India ink and the Negrosine, which is used for capsular stains, or which are capsular stains. The next one, we have our special staining, and this staining demonstrates special features. And our last one is our differential staining that utilizes more than one dye, and it's used to differentiate one organism from another. So I made a table here of the special staining techniques or special stains used inside the microbiology department. So we have first our capsular stains, Common are his, Anthony's, and Tyler and Muir. Spore stains, we have Dorner's, Schaefer and Fulton, Wirtz and Conklin. Our flagellar stains, we have Grace, Fisher and Kohn, and Leifson. Metachromatic granules, we use Alberts, Neisser, Jubensky, Ponder, MB, Lindergren, Burke's technique. Nucleic acid, we use the Filgen technique. For the polar bodies, we use the Weissenstein. And for spirochetes, we use Levadetti, Warthin-Starry, Fontani-Tribundu, and Grumores. And lastly, for Rickettsia or Chlamydia, we use Jimenez, Jamsa, or Machiavello. So let's now proceed to gram stain. So gram stain is the most important stain in microbiology. So I made a table here of the purpose, the reagent, the color of gram-positive or gram-negative bacteria while on the process of gram staining. So our primary stain is our crystal violet, and gram-positive will appear violet or purple. Gram-negative will appear violet and purple, and we use our primary stain for 1 minute. Next, we use our mordant, which is the iodine, for 1 minute, and gram positive appears still violet or purple, and gram negative violet and purple. Next is our decolorizer, which uses acetone alcohol. So in this part, or decolorization is the most critical part in this step because it will interfere or false positive or under decolorization will lead to false positive reaction, and over decolorization will result to false negative reaction. So for our decolorizer, we use the acetone alcohol, and gram positive organism will remain violet or purple, whereas the gram negative will be colorless, and this one or we use the decolorizer quick on and rinse. So our secondary stain or our counter stain, which is the safranin, can stay for 30 seconds, and gram positive bacteria will still remain violet and purple, and gram negative bacteria will absorb the color of the safranin, which will turn red or pink. So I want you guys to also remember that higher forms of organisms like actinomyces, streptomyces, yeast, and molds are reported as gram positive. And all spiral organisms, though hard to stain, are recorded or are reported as gram negative, and Rickettsia, Chlamydia, Mycoplasma, and Ureaplasma cannot be gram stained. Also remember that we have the Hawker's modification of gram stain, and this kind of gram staining is used for fungi, and we use crystal violet as our primary stain, but we add ammonium oxalate on the Hawker's modification of gram stain. So always remember, guys, that the presence of lipid in gram negative makes them soluble to alcohol, leading to decolorization. Again, the presence of lipid in gram-negative bacteria makes them soluble to alcohol, leading to decolorization. So also I made a table here about the possible causes of false positives and false negatives when it comes to gram staining. So let's talk about first the possible causes of false positives wherein gram-negative bacteria becomes gram-positive bacteria. So we have two, under decolorization and thick bacterial smear. Meanwhile, the possible causes for false negatives or false negative reaction wherein gram-positive bacteria becomes gram-negative bacteria. We have three, over decolorization, acidic iodine, and use of penicillin. So always remember, guys, that all cocci are gram-positive except for Branhamella or Maraxella, Acinetobacter, Neisseria, Kingella, and Veillonella. And all bacilli are gram-negative except for Mycobacteria, Bacillus, Clostridium, Corynebacterium, Listeria, Erysipelathrix, Lactobacillus, Crithia, and Rothia. Let's proceed to acid-fast staining, and we use acid-fast staining to stain bacteria that have a high lipid and wax content in their cell walls and do not stain well with traditional bacterial stains. So, in acid-fast staining, guys, remember that steaming process removes mycolic acid temporarily and increasing concentration of phenol and basic friction and prolonging contact of stain with the material. So let's differentiate now the Zeal-Nielsen or hot method from the Kenyon's or cold method. So our primary stain for the Zeal-Nielsen or hot method is our 3-gram carbofixin plus 5% phenol. Our mordant here is the heat or steam, and the decolorizer acid alcohol or it's hydrochloric acid plus ethyl alcohol, and our counter stain here is our methylene blue. So for the result, acid fast organism appear red and non-acid fast organism appear blue. Meanwhile, our canyons or the cold method uses 4 grams carbofluoxetine plus 9% phenol, and our mordant here is tergitol, and our decolorizer still is our acid alcohol, and our counterstain is still methylene blue, or we can also use the malachite green. So for the result, acid fast organisms appear red and non-acid fast organism appear blue. And I want you to take note also, guys, the tergitol or the mordant used in canyons or cold method that addition of wetting agents like tergitol prior to the stain solution, or we can or it's commonly used to facilitate acid fast staining in canyons or cold method. So our standard AFB smear is 2 by 3 centimeters, and the modified acid fast staining or we use this for the staining of the coccidians. We use sulfuric acid plus ethyl alcohol as our decolorizer. And our rule here is all bacteria are non-acid fast except for Mycobacteria, Nocardia, and Rhodococcus. So we also have other methods of acid fast staining. The first one, we have the Pappenheim's method, which differentiates Mycobacterium smegmatis, which is blue, from Mycobacterium tuberculosis, which is red. Next, we have the Baumgarten's method, and we use this acid fast staining to detect TB in tissues, and or it helps differentiate Mycobacterium leprae, which is red, from Mycobacterium tuberculosis, which is blue. And our last method is we have the Fight Farrakos method, and it detects Mycobacterium leprae and uses hematoxylin as its counterstain. So our next part of studying or ways of studying bacteria is also to study their cultural or colonial characteristics in culture or in different kinds of media. So for the first one, we have the types of culture. Number one is our stock culture, which is used for quality control purposes only, and it's stored at negative 20 degrees Celsius to negative 70 degrees Celsius. And meanwhile, our working culture is stored at 2 to 8 degrees Celsius. So we have two types of working culture. The first one is mixed culture, and the second one is our pure culture. So we use mixed culture for two or more organisms, and we use pure culture for identification and AST or antimicrobial susceptibility testing. So let's talk more about our cultural media. So first one, we should differentiate them according to their physical state or consistency. So, our liquid media have no solidifying agent or they contain 0% agar. Examples we have our nutrient broth, our brain heart infusion broth, and alkaline peptone water. The next one, we have our semi-solid media and they contain 0.5 to 1% agar. For example, we have our SIM or sulfide indole motility agar. And next, we have our solid agar that contains 2 to 3% agar. And our examples is our MAC, BAP, etc. And biphasic means they are both solid and liquid. And our examples here, we have the HBBT or human blood bilayer twin agar and Castañeda culture bottle. Next, we should also differentiate them according to their manner of dispensing, and preparation. We have first our plated culture media. Take note of this one, guys. When we do or when we make plated culture media, we first weigh, dissolve, sterilize, dispense. When we prepare tube culture media, we weigh first, dissolve, dispense, and sterilize last. Okay? Always take note of this one. Plated culture media, weigh, dissolve, sterilize, dispense. Tube culture media, we weigh, dissolve, dispense first, and sterilize last. Let's move on to their according to composition. For the first one, we have this synthetic or chemically defined. This means that all components are known to the user and it's mainly for research purposes. Example here is our Siemens citrate agar. The next one, we have our non-synthetic or complex, and it contains of at least one component which is not chemically defined, and it's useful for bacterial isolation. Our example here is our trypticase soy agar. And the last one, we have our tissue culture media, and these are for organisms which cannot grow on free cell media, and they are often used for viruses. And our example here is our HeLa cells, A549, Hep2, Ferrocells, and McCoy's tissue culture. And the last one, we have the according, or we should differentiate them according to their function. So first, we have our simple or basal or supportive or general or purpose culture media or general purpose culture media, and they contain only the necessary nutrition to support bacterial growth. Our examples here are nutrient agar, nutrient broth, trypticase soy agar, and trypticase soy broth. And next is our enrichment media, and they enhance growth of pathogens in a specimen where pathogens is lesser than non-pathogens. Our example there is our APW or alkaline peptone water and selenite F, and we have the tetrathionate broth. The next one, we have our enriched media, and these are incorporated with substances to support fastidious organisms. And our example here is our CAP and our BAP. Next one, we have our selective media that promotes the growth of the desired organism while inhibiting the growth of others. We have our Lowenstein-Janssen or LJ, our cysteine tolerite blood agar CTBA, mannitol salt agar or MSA, and thiosulfate citrate bile salt sucrose agar or TCBS. The next one, we have our differential media, and differential media contains ingredients that allow visualization of metabolic differences between groups or species of bacteria. Our examples there is our MAC, EMB, Hectone enteric agar, Salmonella and Shigella agar. The last one is we have our transport media, and these are designed to preserve the viability of microorganisms in the specimen but not allow multiplication. So our transport media or our examples here are Stewart's, Amy's, Carrie Blair, Transgrow, and Gembeck. So now let's proceed to our biological safety level. So we have first our BSL1, and this is used or this is classified as organisms with no known potential of infecting healthy individuals, and they pose minimal threat to laboratory workers. And the common organisms here are Bacillus subtilis and Mycobacterium gordonii. And our BSL2 are organisms that may pose moderate threat to lab workers. These are associated with LAI or laboratory acquired infections, and these are these are organisms that are often acquired through ingestion, percutaneous and mucous membrane exposure. So our example there is our Salmonella, Shigella, hepatitis B virus, and HIV. And our BSL3, they are high risk organisms with treatment, and they are potential aerosol transmission, or we can also or we can acquire it through aerosol transmission. And our examples there are Mycobacterium tuberculosis, Francisella, Brucella, Rickettsia, and systemic fungi. And the last one, we have the BSL-4. They are high-risk organism with no available treatment, and they are life-threatening diseases handled in research facilities like smallpox and Ebola virus. So always remember, guys, that biological safety level are organisms or different organisms handled in the laboratory, and how can we acquire them. Now let's proceed to biological safety cabinet, and this is a device that encloses a workplace. It sterilizes air with infectious material through filtration using HEPA filters. And HEPA filters, or HEPA, means high efficiency particulate air. Again, HEPA means high efficiency particulate air, and they are classified according to the degree of containment it can afford. So first we have the class 1. It offers the least protection and system is entirely open, and it sterilizes air to be exhausted. So this is mainly for BSL-1. And next we have our class 2 biological safety cabinet, and it sterilizes air to be exhausted and the air that flows over infectious materials. And it is a requirement for laboratory with microbiology section, and these often used for BSL-2 and BSL-3, and also for viral and fungal cultures. So we have two subtypes of class 2. We have class 2A, which is inside the room, and class 2B, which is outside the building. And the last one, we have our class 3, and it provides the most protection. So system is entirely closed here, and the infectious material is handled using rubber gloves attached and sealed to the cabinet. And it's recommended for handling viruses and BSL-4, and it's for BSL-4. So now let's proceed to the methods of microbial control. First, we have the sterilization. So sterilization means the complete destruction and removal of all forms of microbial life, including spores. So first we have, or let's differentiate the physical methods from the chemical methods one. So let's have the physical methods of sterilization first. We have the moist heat, and moist heat requires shorter time, and it's a method of choice for heat stable objects. And we have here the autoclave as an example. It's the fastest and simplest. And the principle of your autoclave is steam under pressure. And we our steam under pressure is use 121 or at 121 degrees Celsius for 15 psi for 15 minutes, and our indicator is Bacillus thermophilus for autoclave. The next one, we have the tendalization, and tendalization is used at 100 degrees Celsius for 30 minutes for 3 consecutive days. And we use the Arnold sterilizer, and the principle is flowing steam. The last one for moist heat is we have inspissation, which is used for 75 to 80 degrees Celsius for 2 hours for 3 consecutive days. And the principle of inspissation is thickening through evaporation. And also take note that inspissation is for media with increased protein, like LJ medium for Mycobacterium tuberculosis. Next part or next type of physical method of sterilization is we have our dry heat, and it requires much longer time, and we often use this for our glasswares. So our example here there, number one, is an oven that we use at 160 to 180 degrees Celsius for 1 to 2 hours, and it's for glasswares, cotton swabs, oils, and powders. And our indicator there is Bacillus subtilis var Niger or var Niger. So the next one, we have our incineration that we use at 870 to 980 degrees Celsius to dispose infectious waste. And we have flaming that is used for inoculating loops and needles, and crenation that is used for burning materials into ashes to control diseases. And next one, we have filtration, and this is done for heat labile objects, and it's used to sterilize culture media, antibiotics, and vaccines. So our first example there is our millipore filter, which is 0.22 in diameter, and it provides 100% sterility. Meanwhile, our HEPA filter removes objects larger than 0.30 and it can filter out viruses. And our last example, or our last physical method of sterilization, is our ionizing radiation. So it is used or exposure to gamma rays and for disposable plastic syringes, or it's used for disposable plastic syringes, catheters, and gloves. Now let's proceed to the chemical methods of sterilization. We have the first one, which is the ethylene oxide, and it's the most commonly used sterilant. It is used in gaseous form for sterilizing heat sensitive objects, and our indicator there is Bacillus subtilis var. Globigii. Number two, we have our formaldehyde vapor and vapor phase hydrogen peroxide, which is used to sterilize our HEPA filter. Next one is our 2% glutaraldehyde. It's sporicidal. It can kill spores in 3 to 10 hours, and it's used for medical equipment. And the last one, we have our peracetic acid. Now let's proceed to disinfection. So disinfection is defined as destruction of pathogens, not necessarily all microorganisms and other spores. So let's also have examples for physical methods and chemical methods. Let's start with physical methods of disinfection. We have pasteurization that remove pathogens in foods, and it sterilizes milk and milk products. So we have three types: our batch method that uses 63 degrees Celsius for 30 minutes, our flash method that is used for 72 degrees Celsius for 15 seconds, and ultra high temperature method, which is used for 140 degrees Celsius for 3 seconds. Next, we have our boiling, which we use at 100 degrees Celsius for 15 to 30 minutes for surgical instruments or medical instruments. And the last one, we have our UV or non ionizing radiation using UV light. Let's now proceed to the chemical methods of disinfection. We have our antiseptics, and antiseptics are chemical germicide for use on the skin or tissue. So our most common here is our 70% alcohol, and we also have our iodophores, chlorhexidine, and hexachlorophene. So while 70% alcohol is the most common, iodophores, which is consists of iodine plus detergent, is considered the best. And we require 2 minutes contact time of ROH and iodine in our skin. And iodine plus isopropyl alcohol is what we called our iodine tincture. Again, 70% alcohol is the most common. Iodophores are consist of iodine plus detergent, and it is the best antiseptic. And we require 2 minutes contact time of alcohol and iodine. And iodine plus isopropyl alcohol is what we call our iodine tincture. Now, our disinfectants, and these are for non-living things. And the examples here are halogens, salts of heavy metals, aldehydes, and quats. And quats are examples of quats are benzalkonium chloride or zephyrin. Always or also take note, guys, that Pseudomonas aeruginosa grown in ammonium acetate media are resistant to quats. Again, Pseudomonas aeruginosa grown in ammonium acetate media are resistant to quats. And the last one is our phenolics. And Sodium hypochlorite or household bleach is our best disinfectant. I didn't add there, so just take note. Sodium hypochlorite or household bleach is our best disinfectant, and the ratio of it is 1 is to 10 or 0.5% bleach for blood spills, and we use vinegar as a substitute if there's no household bleach. Again, sodium hypochlorite or household bleach is our best disinfectant, and we use it at 1 is to 10 ratio or 0.5% bleach for blood spills. And if there's no household bleach available, or if there's no sodium hypochlorite available, we can also use, or we can use vinegar as a substitute. So now let's proceed to the last part of the part one of the microbiology, which is the methods of susceptibility testing. And number one here is our automated system. And it uses, or our example is our Vitek 1 and Vitek 2. These are machine that is used for susceptibility and microbial identification. And we can often see it in our respective laboratory and other research facilities or research institutions. Number two, we have our ETAs, and ETAs are used or alternative for fastidious organisms. So it uses a strip of varying concentrations and of antibiotics along its length. So the positive result here in the ETAs is the ellipse of growth inhibition. So that's why it's E, ellipse of growth inhibition. So associate. And the next one is our D-test, and it detects inducible clindamycin resistance among S. aureus. And the positive reaction or the positive result here is blunting of clindamycin zone to give a D pattern. So in order for you to remember it fast, E-test ellipse and D-test D pattern. Okay, so let's proceed to our tube dilution, and we often use this for MIC, which is the minimal inhibitory concentration, and MBC, which is the minimum bactericidal concentration. So always remember that MIC is the lowest concentration that inhibit bacterial growth, while MBC is the lowest concentration that kills bacteria. And the last one, we have our agar dilution, which is used for our many organisms versus single drug. And this one is the reference method for susceptibility testing of anaerobic bacteria. Again, agar dilution is the reference method for susceptibility of anaerobic bacteria. And the last one, we have our disk diffusion. And if the agar dilution is many organisms versus single drugs, this diffusion is one organism versus multiple drugs. And this is the common susceptibility testing that we use inside the laboratory. So first, we have the Kirby Bauer, or it's the most common method of AST. So we use the Miller-Hinton agar of the MH agar as the required media, and its pH is 7.2 to 7.4, and it's 4 mm, and the storage condition requires that it is aerobic and no CO2. The incubation time is 16 to 18 hours, and the size of antibiotic use here is 6 mm, and the inoculum is 1.5 times 10 to the power of 8 and we use the standard to compare inoculum, which is our zero point 0.5 Macfarlane standard, which contains 99.5 ml of 1% sulfuric acid and 0.5 ml of 1.175% barium chloride. So MHA, or let's go back to MHA, which is the required media, or this is the general media for antibiotic or antimicrobial susceptibility testing. And MHA, or I didn't add there, please take note. MHA plus 2% NaCl is commonly used for Staphylococcus for the detection of MRSA, and MHA plus 5% sheep's blood is often used for Streptococcus. Again, MHA is our general media, or Miller Hinton agar is our general or required media, but MHA plus 2% NaCl is used for Staphylococcus for the detection of MRSA, and MHA plus 5% sheep's blood are used for Streptococcus. So we also have here, or I made a table here about false resistant or false or factors that will lead to false resistance and false sensitive or false sensitivity of the or on antimicrobial susceptibility testing. So we have our false resistant first. We have our heavy inoculum, thick medium, delayed this application, calcium and magnesium, increased moisture, and prolonged incubation. And false sensitive or very light inoculum, thin medium, very dry agar, acidic pH, mainly due to the antimicrobials like tetracycline, methicillin, and novobiocin, and alkaline pH caused by aminoglycosides and erythromycin will lead or may lead to false sensitive reaction. So I have added there some notes. So guys, please take note that 150 plate is used for no more than 12 discs, and our 100 plate is no more than 5 discs. Also remember, or also take note, that 24 is the distance of disc from the center, and 50 is the distance between the two antibiotic discs. And the discs are pressed firmly to ensure contact with the agar, and within 15 minutes of displacement, plates are inverted and they are placed in a 35 degrees Celsius ambient air incubator for 16 to 18 hours. So we use ruler or caliper to measure the zone of inhibition, and we always ignore swarming. Always remember this one, to ignore swarming when we are measuring the zone of inhibition of the or for the antimicrobial susceptibility testing. So for the last one of our part one, I have listed there some of the antimicrobial agents and their uses. So the first one, we have our cell wall inhibitors. You can see there we have beta lactams, glycopeptides, cycloserine, and bacitracine. Our cell membrane inhibitors, we have our colistin, polymyxins, amphotericin B, and nystatin. Our protein inhibitors, we have aminoglycosides, tetracycline, chloramphenicol, and macrolide. And our nucleic acid inhibitors, we have our sulfonamides, trimethoprim, quinolones, rifampicin, and nitrofurantoin. So always remember, guys, that bacteriostatic antimicrobial agents are examples of bacteriostatic antimicrobial agents are chloramphenicol, erythromycin, clindamycin, and sulfonamides. And bactericidal, we have vancomycin, aminoglycosides, beta lactams, and quinolones. So that's all, guys, for microbiology part one. Thank you for listening.